to Virology.
Sure. Welcome to Q&A with A&V. Hey, everybody, I'm Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you today?
I'm well. So you want to change, what is it on the logo? It says right now, Q&A with A&V, answering your
COVID-19 questions. It should be answering your virology questions. Shouldn't it be more expansive?
Well, let's ask our participants here. We have 135. What do you think? Should we change the title to Answering Your Virology Questions? And then we would probably not get a lot of people. Anyway, let us know what you think.
You think, okay. Well, people sometimes ask non-virology questions, non-COVID questions.
So Feruza says hi to you, Amy. Here.
Hello, Feruza.
I should put that up. Here you go, so you can see her. She's a virology PhD student in Canada. Your favorite country, right?
My favorite country?
I'm joking.
I know. I'm thinking. Well, it doesn't have Paris.
Nope. All right. So we have we have a major announcement.
Oh, we do.
Yeah, on Monday we achieved 100,000 subscribers on YouTube.
Yay!
Isn't that amazing, folks? And
That's
so
great.
thanks to all of you who have followed and everybody else. It's so funny because at the beginning of 2020, I think I was just over 20,000. And
well, pandemic can do it for you.
So the response is keep COVID and maybe say COVID and virology questions. They want to say keep COVID for another 6 months.
Okay, that's fine.
And some people say leave it as it is. So
okay, that's fine.
You know. So I just am very kind of humbled that 100,000 people want to watch science. And so that's great. Now we have quite a platform. And we can move forward, right, Amy? What do you think of 100,000 subscribers, Amy?
It's 100,000.
Okay. Amy is her usual
I'm not clear.
Straightforward self. Yeah.
I mean, I think it's great, but, it's 100,000. So it's good. It's just, I think it's, it can grow larger. I'm sure it will get bigger.
Yes, I have a haircut, folks. Yesterday, it was getting out of control, and people were saying I had bedhead when I didn't. I would never appear.
It's actually not so bad to have bedhead. Not that I have it, but it's not bad.
Amy is a bit blurred. Yeah, I don't know why. She's on an autofocus camera, so maybe it's focusing on your microphone. I don't know. Can you push it down a tad? The microphone, I mean, not the camera.
Sure. How's it?
Yeah, it's a little better. Bedhead is when you roll out of bed and your hair is all messed up, right?
I guess.
So I don't do video stuff rolling out of bed. I take a shower always.
Well, everybody takes a shower before they put on their clothes.
I don't think everybody does. I think there are people who do not.
Well, okay.
All right. So here's our first question. If all COVID cases were symptomatic, do you think we would still have a worldwide pandemic?
No.
Because SARS-1 was mainly
all symptomatic.
All symptomatic, and peak of shedding was at the peak of disease, and over half of the people were in the hospital. So,
yeah, but she doesn't ask about whether or not what the peak of disease has to do with it. If everybody were symptomatic, we'd be done.
One and done, yeah. Another one from Elena or Elena. I don't know where you put the accent. If Kathy were here, she'd correct me. But
well, she wouldn't know. You could be either way.
You can say it either way, yeah. What's the main difference between the mRNA vaccines? Is it basically the types of lipid used?
Basically.
Yeah. will the antivirals have to be given in a cocktail to avoid resistance?
Absolutely.
But they will not be initially because they're only being tested singly, right? Which is how you do it. You test them singly to make sure they're okay. Then you have to do combination trials, which means the companies that are making them. Did you know, Amy, Pfizer's making a protease inhibitor?
Yes, I knew.
And Merck has the polymerase drug, and I don't know what the third one is. Do you know, Amy?
No, not offhand.
Okay, here we go.
But, I mean, it doesn't take rocket science to figure out that you needed to do combinations. You just had to look at our experience with HIV and how fast we got resistance to AZT. So, when they started, so molnupiravir came out of the Emory's clinic, chemistry lab that was associated with the flu center, right? The influenza center. So it's been sitting there for many years.
It has been. It's too bad, right? We should have had it.
Well, yeah, but my point is it's been sitting there for many years. And so they should have known that they should have, if they wanted to advance it as a flu drug, even still, you should have combined it with something else.
Yeah.
And then you would already be halfway there, right? Because all you really need to know is if it's going to be toxic.
Yeah, and then you could have done a phase 2, right, at the beginning of the outbreak.
Yeah, or whatever. Even if you did a phase 2 at the beginning of this year, my point really is that you would have known that you would have been already in position to make it a cocktail. Of course. That's my point.
The thing is, Amy, as you may agree with, we didn't put billions of dollars into antivirals. We put them into vaccines, right?
Yeah, but it doesn't even matter that we put billions of dollars into vaccines. What I'm basically saying is for any virus that you want to develop an antiviral, whether or not it's flu, it's EV68, it's, measles, you automatically have to think that you're going to need a combination. You should just start off with the idea that you need a combination. So...
Well, that's not how they're doing it, as you know.
Well, yeah, but... As you know, there's this idea that we're going to have a pandemic preparedness center, right? This country is going to fund pandemic preparedness, right? And so wouldn't you say if you're going to fund pandemic preparedness and you're going to have a branch that does antivirals, that you should automatically start off with the assumption that you're going to need a combination therapy? So that's what I'm saying.
Yeah, I agree with you, but it's not done that way anyway. And BJR2, thank you for your contribution. 2 PharmDs in San Francisco awaiting your Q&A with worm on tongue. Now, I don't know what worm on tongue means. Do you know, Amy?
No.
Okay, some kind of anticipation. Why do the lipid nanoparticle chemicals concentrate in ovaries? Have you seen this report, Amy?
No.
So, Robert, this is the problem. This is a leaked report, which was found in Canada or something. I don't know what it's from. We have no idea the provenance of it. But they showed that 0.09% of input, an experimental animal, I would think, local of mRNA vaccines, lipid nanoparticles, home to the ovaries. I have no idea what that means and whether it's related to menstrual irregularities. I don't know what that means either. This is a big problem in looking at information that hasn't been vetted yet.
Wait a minute. It was done so this is from animals? Like with a normal dose, or where they do the biotissue availability studies where they give 4 million times the dose?
Hold on. Here's the story. A leaked document that was a pharmacokinetics study. They put a lot of lipid nanoparticles into an animal and they see where it goes and for how long. It's now being used by a Canadian immunologist to spread the rumor that the nanoparticles are accumulating in ovaries. So it's a rumor. And this is the kind of...
So let me get this straight. They put in 4 million times the amount of particles that you and I are ever going to see. It goes everywhere. because that's what happens when you put in 4 million times the amount of particles that you
Yes,
ever need.
that's right.
And then we're going to make a conclusion that it's toxic. Is that how this works?
Yeah, you got it pretty much, Amy.
Okay. All right. This is a good plan. I
I
like
think
this.
this is a
This is a
ploy
good
by
plan.
people to discourage people from being vaccinated because they don't like the idea. So, Robert, not good, not good. I see the Moonshot project got a lot more funding. Could such an open source antiviral help solve the lack of vaccines in the third world, or would it take so long? What do you think, Amy?
Could an open source, what's an open source for antiviral mean?
When a lot of people participate in its development. Because open source software, everybody works on it, right? The code is there online. Everybody works on it, contributes. I don't see that happening with an antiviral. Do you?
It's too expensive to develop, and it's too dangerous. Because you make one mistake and somebody could die, right? I mean, not for like even if the drug is safe, you give them it in the wrong administrated form. You give it IV instead of oral. You know, it's not packaged properly. I mean, there's a lot that goes into drugs, like how you would dissolve it, blah blah blah. I mean, I don't think it's all that easy.
No, I agree.
So.
Just finished listening to Virology Live 3. Loved listening to you both. Might it be possible someday to silence the extra 21st chromosome in people with Downs?
Silence how?
I don't know. Could you CRISPR it out? But the damage is already done, right?
Yeah, how are you going to CRISPR it out? So you want to CRISPR it out when it's detected in an amnio after you've become like a gazillion cells? You'd have to CRISPR it out at the stems, at the germline.
Yeah. And you wouldn't know at that point.
No.
So I don't think that's feasible. During the winter virology course, I started making a list of abbreviations. Glad to share. Well, we had some people here who were going to make a glossary. I sent them actually the glossary from the book, and they were going to make a document. But we'd love to have some starting document that online that we could do open source, an open source virology glossary. That would be fun.
Would be good.
What's the relationship between lymphocyte concentration in myocardium, myocardial damage, and later fibrosis with compromised cardiac output? I doubt anyone knows, because who's going to do biopsies of that to find out, right? That's what you need to do.
How are you going to do the biopsy?
You know, the sternum, Amy, you put a needle and you kind of point it upwards.
Like
right?
what we're going to do in a couple of weeks for the mice?
Yeah.
Yeah, but the mice will be dead.
Yeah, I don't think you can do that in people. So I think if you're talking about myocarditis, either in COVID or in vaccination, I think it's inflammatory.
Or in general.
It's inflammatory driven. Yeah, in general. And the problem is also, bless you. Oh, no, you're not sneezing.
No, excuse me. What? The problem in general is what?
For trisomy, which is someone distracted me with a trisomy, is that the kid is born and they have their issues already, developmental and cognitive and so forth, and taking out the extra chromosome is not guaranteed to fix it, right?
Right. But I don't know that you would. I don't know that she meant that you would silence it after it was born, or you would do it in utero. But the thing is that how would you know? It's not at the 8-stage cells. It's not at the 8-cell stage that you would figure it out. It's not that early on. It's much later, and so there's no way you could do it.
Right. Michael DeSanti's cancer breakthrough involves antibiotics to treat cancer.
Okay.
Seriously? And what's the logic there?
It inhibits translation elongation.
In eukaryotic cells?
Yeah, it's not the most successful or well thought out plan, but there's lots of therapies that aren't the most successful and well thought out plan.
It sounds like a clinical plan that's not basic science thought through. As for COVID, wouldn't antibiotics that suppress protein synthesis also suppress synthesis of antibodies? Yeah, they would suppress everything, right, Amy?
Yeah, you don't get. I mean, I don't know as why it would. If you give antibiotics independent of any infection, you're suppressing protein synthesis.
That's right.
Doesn't matter. You're targeting the host ribosome
I don't think it's a
or
good strategy.
translational machinery. That's what I'm saying. It's
Yeah.
not a good. That's what I just finished saying. It's not a good strategy.
For COVID, also, it's not a good strategy, right?
It's not a strategy whatsoever.
I liked hearing about the vaccine for malaria, so probably we'll talk about it on TWIP on Friday. So tune in. I think TWIP live stream would be fun, don't you think, Amy?
Yeah. Are you planning on getting a word in?
Why do you say that?
Because Dixon likes to tell a lot of stories. He's very interesting. He's a great storyteller. And I think that when somebody asks him to tell one thing, it's just going to snowball.
Yeah. I don't mind Dixon talking. It's fine.
I didn't say I minded him. I just said you would never discuss the paper or the topic. You would start off at one place and you would end probably 3 steps away from another place.
Thank you, Tom, for your contribution. No problem missing, obviously. The very young are at high risk for serious disease. What data do we have that supports this? Well, where did you get that conclusion, Peter J.D.?
They're not, actually.
They're not. They can get serious, but they're disease. They can get MISC. By the way, Friday on TWIV, we have 2 experts on MISC. They can get serious disease, but they are not at high risk for it. It's not 0, though. This is not from the incubator because it's 8 p.m. and so we go till 10. I go to 10. And then the next train would probably be 10:15, which would get me home at 11-ish. I don't really want to get home that late. But maybe sometime I'll do it from the incubator. It's fine with me. I'm sorry, Amy. I don't have a Brooklyn accent. You're right. Have either of you seen or heard of the study that showed the influenza vaccine lowered the risk of dementia in older folks?
I have not. Have you seen that?
No, I have not. I'm trying to think of why it would. So there is a paper from a couple of years ago. So both influenza vaccines and pneumonia vaccines are tied to a lower risk of Alzheimer's dementia, but it's an association. We don't know what that means.
Where was it published?
Oh, you want to know the real thing, right?
Yeah, I want to know if this is a believable thing or some fakakta idea.
By the way, I used that word on the course today, fakakta, and I gave you credit for it.
Oh, really?
Yeah, okay. I said it's a word that Amy often uses.
Yeah, for things that are screwed up.
Yeah, I forgot what I mentioned it for now. Anyway, this is a report at the Alzheimer's Association International Conference in 2020. Seasonal flu vaccine may reduce incidence of Alzheimer's dementia. I don't know if it ever got published. So.
Well, I just want to know, I highly doubt any virologist did this. And
you would probably be right, yeah.
And it's in this, yeah. Okay, move on.
And Gribbler found it also, yeah.
Move on.
Jack says, do the seasonal fluctuations in the incidence of COVID reflect intrinsic properties of the virus, or are they just due to human behavior? Probably both and more, right?
Well, what's the intrinsic property of the virus?
If I explain it, you're going to say, where does he say that? Right? So I'm not going to explain what I think he's meaning. How many months have we been doing this live stream? I got it by now. I figured it out. I'm no dummy. Well, I am a dummy, but I got Amy figured out to a certain extent.
It took a little under a year, about 11 months for you to figure it out.
Do you know we've been
Okay.
doing this for over a year?
For 11 months. We started in November, I thought. Okay, maybe it was October. But yeah, okay, for a year? Wow. That's
Yeah,
a long
almost. It's
time.
not a year yet. It's not a year. You're right. Yeah.
It's human behavior, number one, right? And then it's whether or not you have homotypic immunity, right?
Yeah, but I think this, the seasonality, the weather conditions that impact some viruses, humidity, temperature,
Yeah, but that
maybe
has to do with the size of the droplets, right?
probably. Yeah.
That's not an intrinsic property of the virus. That's a reflection of
Yeah.
how much water is in the droplet that's coming out of you.
Okay. If vaccinated, is it possible to tell if you were infected with COVID before your vaccine, or can it only indicate that you've had an infection which may have happened after getting vaccinated?
No, you can do a nucleic capsid antigen test.
Which would tell you if you're currently infected, right?
Would tell you either one, right? One is I believe that there's one that looks for nucleic capsid antigen, and one that's an antibody test against nucleic capsid. One would tell you if you've had a previous history, right? The antibody test, and if you had the antigen, then you would be currently infected.
But it wouldn't tell you before or after the vaccine, though.
What are you talking about? Well,
so the question is, can you tell if you're infected before
the vaccine? Yes, you do an antibody test for nucleic capsid. You get vaccinated, you get a test for nucleic capsid.
Yeah, but if you were infected after the vaccine, you'd still be nucleic capsid positive. That's what I'm saying.
Okay. But or can it only indicate if you had an infection which may have happened after getting vaccinated? Any test is never going to really. Well, actually, the antigen test will tell you if you got an infection after you got vaccinated. So if you don't have an antigen and you have an antibody and you got vaccinated, right, you can conclude that the infection occurred before you're vaccinated, right?
Yeah, but an antigen positive means you're currently infected. But what if it's months after both vaccination and infection? You're not going to have any antigen. That's my point.
Yeah, I got it.
Okay. Here's a question for you, Amy. How long after infection until the memory B cells start making?
6 months at least.
The first exposure?
Yeah.
No, they can make antibodies before then.
Memory B cells take like at least 6 months.
First exposure, right?
Yeah.
Okay, not
memory
the
B cell,
not the
B cell, not the CD20 positive B cell. The next step takes like 6 months.
Now here we have lots of here's answers to our question. What should we call this? It says it doesn't matter as long as it says A&V.
That's cute.
See that?
It's good. It's nice. It's good.
Are there any special conditions which would promote or favor recombination of viral genes in a co-infected cell? Would it produce a new strain faster than base-by-base mutation?
So if you had a lot of homology.
Two very similar viruses?
Mm-hmm. Yeah, or two very similar regions, right? You don't need to have a whole virus that's similar, right?
Right.
You make so, I mean, you could, yeah.
And it could make a new strain faster than base-by-base mutation, right? Because it would happen immediately. So
well, it depends, right? It depends on where the base is, right? Yeah. You only really need one base change to make a new virus, right? Or something or other, right? If you get lucky.
Here's something. Peter says, whatever Amy wants to talk about. You got fans, dude. You got fans. H-Sue. Hello, H-Sue. I heard some talk about H5N8 avian flu. Is it possible to predict human-to-human transmission by looking at the genome sequences?
No.
Nope. Can't do it.
But if somebody could hold up the envelope to their head and do it, I'd like to meet them.
I don't have an envelope on my desk. Yeah, but Amy, can you hold up a tube of virus to your head and ask if it's going to transmit better?
No, I
Okay.
cannot. I need an interpreter, and they like to be wined and dined first. The virus likes to be wined and dined, and I'm out of wine. They
Yeah,
request
COVID.
what they request, you got to cater to what you want it to do.
Yeah, so there's mixed thoughts. I think we have to keep COVID in the title for a while, Amy.
Okay.
Do you know how many days after a possible exposure to SARS-CoV-2 does it take before you shed virus? Is it still 10 to 14? No, I don't think it's 10 to 14.
I thought it was 5.
You could shed as soon as 5, but I think it could take as long as 14 days, right? Maybe 10 to 14 is the average, but it could start at 5 days post-infection, yeah. Now, whenever we do with numbers, folks, sometimes we make a mistake like we did last time, and you guys get all bent out of shape. Just chill. I
I
think...
like this.
What?
Just poor Dixon, just saying Amy's social skills suck. Like that. My social skills suck. Was I picking on Dixon?
No, she wasn't picking on Dixon. She was just saying he likes to talk, and I agree with
Well,
that.
I think he has interesting stories to share, and I think he's a great teacher in sharing them, and that's the whole point of what his personality is.
Well, what you folks don't know, and not you folks, but it was one person. When Dixon used to come in, Amy and he used to chat often, daily. She would stop by his desk and chat. They liked each other, right?
Yeah, I miss Dixon. I miss chatting with him. He always had interesting stories. And I don't think it's a bad thing that you start off with one thing and you end up on a completely different topic. I think that's the way it works. That's how you learn, and you have interesting dynamic conversations.
That's right.
So I think a live stream with Dixon. If you and Dixon did a live stream, it would be phenomenal because Dixon would start off with this and then he'd say, but I really, had this wonderful time when I went on this trip to Kerala and we went to visit the things and then, we ate this palm sugar, but it's not really sugar. It's, whatever. And then, I think don't you think that that's how Dixon is?
I could get him to do a live stream. I just have to bring him to the incubator. We'll both sit down at the table, and we'll stream it. He would have a blast. He would love it.
Yeah, I think so.
He would.
And he would never tell the same story twice. He's got a plethora of stories, right?
Here's a non-COVID question. Do we know why resistance to immunity hasn't arisen in poliovirus in the time we've been watching it? Presumably the existence of 3 serotypes says it did in the past.
So it still exists at 3 serotypes. We just don't have 3 serotypes circulating anymore. And actually, we do know why there's not resistance to immunity, right? Isn't that what Kara Burns' paper says? Right, partly?
Yeah,
she had antigenic drift, right? And they had multiple sites that were varied, right? And then at one point there was one too many variations and it didn't allow for receptor binding. And so that virus died off, right?
Yeah. So polio and measles are similar in that they just don't vary because apparently the proteins can't handle it architecturally.
Well, the virus, the particle can't handle it architecturally, but they do vary because you can see them vary.
Yes, of course.
Right?
But they don't vary. They don't vary to not be neutralized. Very rarely. It has happened twice. There are 2 examples.
Yeah.
Finland and the Congo.
Yeah.
2 examples of polio antigenic escape. Yeah.
Yeah.
So it's not common.
It's right. But for measles, it's 7 out of 8, right? 7 out of 8 residues is fine. But when you make that 8th change and you alter receptor usage, so it no longer uses, SLAM, because I think CD46 is the receptor only for the attenuated strain, right?
Mm-hmm. Yep.
So that paper that we did on Twiv, where you didn't think it was good until it, you didn't like it until we like you realized it was all because he wanted to make it the oncolytic virus. Remember that?
Yeah. Yes, I got it in the end. Took me.
Yeah,
Sometimes it takes me a while, and that you don't like that. You're impatient, but I have my
yeah,
pace.
but that whole point was right that they saw 7 out of 8 changes, and then that eighth change couldn't be tolerated anymore. Receptor couldn't bind. Blah blah
Yeah.
blah. Right.
Yep. Yep. Amy, someone's asked you, what about a schmagegi? Do
What
you ever
about a
use that?
schmegeg?
What is a schmagegi, Amy?
A schmegegi is like someone who's being super silly. It's kind of the easiest way to
Oh,
think about it.
so I'm never a schmagegi, right?
No.
Good.
Why would you? Yeah, not a schmegegi.
Amy is an unassuming star. Look at that.
That's cute. That's very
You need
nice.
to
Thank
you
you.
need to take these things when you're having down days and just run with them.
And I'm having down days. How about down months?
You have to remember that people love you.
Okay.
Why does COVID-19 trigger type 1 diabetes? My cousin is in the ICU with COVID, was diagnosed, 30 years old, healthy. I don't know why. It may be an inflammatory trigger, right, Amy?
There's some indication. So Ben Tenuver has a paper about talking about how it can kill beta cells in the pancreas. I'm not sure if it directly does it or why. I haven't read the paper yet.
Is there any reason to believe that obesity would inhibit a robust immune response after 2 doses of an mRNA vaccine?
Obesity is an inflammatory disease.
So it
It,
shouldn't inhibit it?
it would. That's why it would. It's because it's an inflammatory disease.
Yeah, but when you get vaccinated, you make an inflammatory reaction.
Yeah, but it's of a
So,
different magnitude. It's like having the M1 cells versus the M2 macrophages.
okay.
And
Did you
that
get
work actually was done by Ira Tabus here and
uh-huh.
Michael Karen. And Michael Karen at UCLA, where they looked at, different triggers for like obesity and inflammation, like downstream of type TLR4.
Yeah. Do you get a plaque? I think so. But as I told Amy today, probably they'll forget about me and not send the plaque. And
They're not going to forget about you. First of all, you know Jack. So Jack cannot forget about you. Plus, isn't there Google Sync this weekend?
yeah, it should be on Sunday.
Yeah,
When
you should go and tell the
I will
Jack you
go.
want your plaque and you want and we want to be able to get the super chats.
Jack is one of the earliest employees of Google, and I got to know him in this pandemic. When I get the plaque, I will show it here, of course, and we'll put
It's
it
an
in the
actual
incubator.
plaque.
I think
It's
it's a
not
real.
thinking. It's not just a little thing on the website. It's an actual plaque.
I think so.
I thought it was just a doohickey that they put on your website.
It says creator awards are given at YouTube's discretion, and we only recognize creators that have played by the rules. One guy who got 100,000 subscribers got an email from YouTube saying the company would not give him a plaque due to the nature of his content. Do I have to apply for this darn thing? Do you get a plaque for 100,000 subs? Pass a specific milestone. When you reach 100,000, you get silver. And gold when you reach 1 million. We're going to hit 1 million one day, folks.
Yeah, but I'm still concerned. I'm still confused. It's a plaque. They send it in the mail.
I believe so, and we're going to hang it in the incubator.
Well, then how do you know it's not in the mail?
Because they only hit 100,000 on Monday. You think they're watching out just for me to give me my plaque?
Do you think you're the only person who hit 100,000 on Monday, or do you think a gazillion people hit 100,000 on Monday?
A gazillion people. I don't know about that.
I don't know, but do you think more than 2?
Yeah, probably. There are a lot of people on YouTube.
Okay, so, and today is only Wednesday, so if they actually put it in the mail, you really think it got here by today?
All right, we'll see, Amy.
Okay, I'm just confused.
Do you think the 1890 pandemic was caused by flu, or you think it might have been a common cold coronas?
Flu.
I've heard, I've heard, but the serological data suggests that it was influenza.
I thought that, yeah.
Concern about vaccine is in the needle hitting the bone or outside the deltoid. What happens if it does? Does the RNA go into the bone? If it hits the bone, you're going to feel it and you're going to hurt. You're going to be sad. Yeah, a little of the
You're going to have
inoculum
a
will go in the
huge
bone.
bruise. No, it goes onto the bone. The outer part of the bone is not really alive.
So you can't
inner
possibly
part of
go
the
into
bone.
it by just a stab of a needle.
Not unless you're breaking the bone or putting
Yeah.
a bone chip.
Thank you, H. for your contribution. Really appreciate it. Come visit one day the incubator. It's getting there. It's not done yet. Today, the exciting news, other exciting news, more furniture is coming tomorrow.
Did you get a time?
I didn't get a time.
Well, maybe they're loading up the truck tomorrow.
We are getting the opposite of clarity on vaccines. This makes me conclude that what we need is to find the best indicators of immunity and test for them. Yes, the correlates of protection. Absolutely. Because as vaccines are licensed, then it becomes harder to test new ones. So then you just ask, do they make this X level of antibodies that we know is the correlate of protection? And you could use that for licensure, but we don't have that. Yes.
Oh, Dixon has an art show at Fordham.
How did you find this out?
Dixon emailed me his art show at Fordham.
Oh, that's cool.
Yeah, I'm super excited. Look. See, doesn't that look familiar, that pattern? Like the one that's hanging in your
Oh,
office?
yeah, I have one of those images in my office, yeah.
Yeah.
Oh, how cool.
Super
Very good,
cool.
Dixon.
I wonder if we should go, if we can go.
Colopy is doing their algebra homework right now. Cool.
Excellent. Y equals mx plus b. Oh, wait, that's a graph.
Does culturing cells on plates versus suspension change virus infection properties?
Yes.
Yeah, you now.
Did he not listen to the TWIV where we discussed the flat and the round?
Oh, yeah. Well, you were
influenza?
on that one. You were on that one, weren't you?
Yes, I was. And I
Yes.
still don't think that there's any biological significance to it because at the end of the day, you're in a negative pressure lung, right?
Mm-hmm.
So how does pressure, change everything?
That's a good question, though.
What?
Yeah, just flat versus round matter. You're going to have more surface area when you're in suspension, right? But we used to do infections in suspensions just because we wanted to get a lot of cells. And I always thought the yield was different in a suspension culture of polio anyway.
Well, yeah, you're going to have a higher yield for sure because you have 6 surfaces, right, for the virus to bind to. On a flat plate, you only have 1 surface. It's
Yeah.
less efficient.
What do I think of triple E? Equine encephalitis. Eastern equine encephalitis virus, which is a toga virus. We've talked about that on TWIV not too long ago because the CDC released its survey of arboviruses, and this was one of them. Yeah, it's big in Massachusetts, for sure, but it happens in other states as well. Have you guys considered the historical importance of TWIV? No, I have not. Imagine
Seriously?
a similar record. Yeah, I have not.
When you say this is my gift to humanity, and you say what other thing have you told me? That's you've considered the historical importance of TWIV for sure.
Well, that's assuming it stays around so that people can consult it, right?
Well, this media probably it's probably like movies. You went from film to VCR to DVD, and now we stream, right? But you can still get Gone with the Wind that was made in 1930.
So, Amy, long after I'm gone, they'll still be able to get TWIV?
For sure. You're willing it to me, right? It's
Sure.
my inheritance, so
Yeah, you gotta.
don't screw it up.
Yeah, but you have to figure out who to take care of because you're not going to be around forever, even though you're a young whippersnapper.
Don't worry, I'll figure it out. I'm not worried.
So
I'm
we have
pretty
been
smart.
documenting, the entire pandemic like no one else has, I think. Yeah, so you could go and listen to all the episodes. That's cool. What determines whether coronaviruses can cross the blood-brain barrier to directly cause brain pathology or not? This is for Amy. She's into this sort of thing, right?
I'm waiting for a coronavirus to cross the blood-brain barrier.
I knew Amy was going to say that.
I'm waiting for one of them to do it. So actually what determines whether or not, it depends on how you want to cross the blood-brain barrier. So some viruses, like West Nile, they make the blood-brain barrier leaky, right? That's what Diamond showed.
Mm-hmm.
And then others use the Trojan horse, like in stuff like EMCV, right? They go through infecting macrophages that can go in and out, right?
Yes.
But
Yep.
so far, neither of those have occurred for SARS-CoV-2. And then there's the stuff like that you and I are most interested in, the ones that bind directly to the surface of neurons, get internalized, and then retrograde in.
Yep.
Right. But once again, no data supports that that coronas enter or cross the blood-brain barrier.
Steve wants to know, how is lovely Amy today? How is lovely Amy?
It's just.
Okay. Taylor wants to know, why do viral vector vaccines need to enter the nucleus, but mRNA vaccines only need to enter the cytoplasm? What a great question.
Viral vector, like adeno or DNA viruses, they need to be synthesized into RNA. They need to be transcribed, whereas mRNA can automatically engage the ribosome that sits in the cytoplasm.
And this process called transcription that Amy refers to in the nucleus, that's where the enzymes are. They're not in the cytoplasm.
No.
A subtitle for this episode, Amy, Personal Hygiene with A&V.
No. But it's a good try.
What do you think about the report in the Telegraph about DARPA getting a proposal in 2018 to fund creation of artificial coronas? So Powell, I actually was asked to comment on that proposal. It's a grant proposal from EcoHealth Alliance, and it basically has the same experiments proposed in it that...
I don't like the term creation of artificial coronaviruses.
Well, they are artificial in the sense that they don't exist in nature, right? You're taking a corona and you're modifying it so no such virus exists.
Most things that you do in a lab don't exist in nature.
Okay.
Do you think soluble receptor PVR resistant variants are there in nature?
Right. So Powell is in a lab, it looks like, so maybe he understands this. But let me tell you about this proposal. So it contains experiments. There are two kinds of experiments in it that have been proposed elsewhere. One of them is to take WIV1, which is a bat SARS-like coronavirus, SARS-related, SARS-1, and put spike genes from other bat SARS-related coronaviruses in it and see if they reproduce in cells with ACE2 as a receptor, okay? So that's been done. And then the other experiment...
proposal.
Yeah. And then it's not even clear this got funded. And then the other one is they wanted to change the furin site and add furin sites to coronaviruses and see what would happen, which is fine also. I don't see that I told the reporter who was talking to me about it, I don't see why there's anything unusual about that. First of all, we don't even know if it was done. We don't know what happened. But even if they did do the experiments, that's fine. I don't have a problem with those. So I think they're fine experiments. I don't think they're dangerous. And remember, it's SARS-1. So this couldn't have led to SARS-2.
Doesn't matter whether or not it's SARS-1 or SARS-2. It doesn't really make a difference. I don't like the fact that the way it's phrased is a negative.
Yeah, I understand.
You're already on the defensive. It's a negative.
Of course.
and it's phrased like they're out to do something
Yeah.
that is harmful and unethical and stuff. And
Yeah. Yeah.
I
Yeah.
don't think that is a good tone. So I listened to this podcast this afternoon.
No, don't tell me Andy Slavitt, please.
No, but the guy has been on Slavitt, but that's not my point. So he talks about how he goes on to like Fox News and whatever the more conservative version of Fox News is, which is like news, power
Hmm.
news. Do you know what I'm talking about? There's apparently something that's more conservative than Fox News.
I don't know. I'm not aware. No.
But apparently there is, I'm telling you.
Okay.
He goes on
Okay.
there. And they say to him, you know, people want to tell you how to raise their kids, and because they want to tell you that you have to put your kid in a mask. And he says, he steps back and he rephrases it and he answers, as a dad, I want my kid to be most safe. And if it's wearing, I don't see the problem, where wearing a mask would make my kid unsafe. Like, he does it, like, they're trying to make my kid safe. Why are you against making my kid safe? So they're trying to understand how we get disease. Why do you want to
Yeah.
make it a negative?
Well, I think people don't understand the basis for these experiments, and that's part of the problem, right?
Yeah. Oh yeah, it's Newsmax. This James Madison person knows what it is. It's Fox News on steroids. Yeah, it's Newsmax. Never heard of it
Okay.
before.
Speaking of podcasts, I was on a podcast. Was it this week? I can't remember. I think it was Monday. Jillian Michaels, Keeping It Real. So Jillian Michaels is a fitness person, right? And
Yeah.
she has this pod called Keeping It Real. So she said, I listened to Lex Friedman. She said, I love Lex Friedman. I listened to all 3 hours and 19 minutes of your podcast, and I loved it. And I wanted to get you on here and talk some more. So check it out. I think it was pretty funny.
I think it's great, but I question her taste. All right,
What,
it's eighth grade.
wanting
No,
me on the pod?
loving Lex, the man who doesn't
Oh,
realize
okay.
he's a big bullshitter who thinks he's not. But 8:47, so 2 more, and then I have to go plate out my cells and finish the grant.
Is the equivalent of Typhoid Mary possible with COVID? COVID Charlie, COVID Karen. How about COVID Amy? No. I don't think that you could shed SARS-CoV-2 for years and years and be infectious and transmit to others, right, Amy?
I don't know. Maybe an immune compromised person who doesn't really develop symptoms well, just like the immune compromised person who got polio who got
Could be.
OPV does, right?
Yeah. All right, here's one for you. What happens to deltoid muscle cells that take up mRNA vaccine and express spike? Are they killed by CD8 T cells?
They could, but most likely are not.
Yeah, because there wouldn't be any. If you're not immunized, there wouldn't be any virus-specific T cells around, right?
Right.
Yep. I have a sense, Amy, tonight we have good, calm questions for the most part. Would you agree? The tenor of tonight's questioning is better.
Sure.
With the exception of one or two that you didn't like.
Well, there's there's less people who yeah.
There's less contentiousness. People seem to be calm tonight.
Which is good.
Although that could change very quickly.
Except for a few people who needed to comment on my social skills and various other things. But they're not the only people who've commented on my social skills, which usually ends in, she lacks social skills, which I usually and wish to remind them, if that is what they're writing, they lack social skills.
No, I'm sorry.
It's okay.
I get emails. I get hate emails too, Amy.
It's okay. And people don't really always understand, like, I'm not picking on Dixon. I'm actually like saying that Dixon's fabulous, right?
Yeah.
I love talking to him. I learned a great deal, not just about science, but life and art and various other things, right? And I think, as we talk about when we write grants, like I find that you should work on things that you're curious in, and you shouldn't always have to start off your grant with this grant is going to test the hypothesis of there's a receptor on the surface of the cell, right?
Yeah, here's one for you. Did anyone read the article about sampling of wastewater plant making judgment on how a population is doing based off of that sample? Well, it's been going on for a while now. They've been looking for virus in wastewater, and they can anticipate when there are outbreaks and what viruses, what variants are circulating and so forth. Right?
Yeah,
Is
for
that
sure.
correct?
Yeah, that's
I
what
have
Davita does, right?
Yep.
She did.
I have been on YouTube since it began. I have clicked on the subscription button only twice, and only one time, not by mistake. It was to you, February 2020. Oh, thank you so much. That's very
cute.
kind, right?
That's nice.
Sylvia,
Excellent.
thank you for your contribution. Really appreciate it. Let's find one more question for Amy here. In spite of it being loss of function for the virus, does attenuation for making vaccines fall into the realm of gain of function?
It doesn't really confer a new property. It weakens a property.
Yeah, that's right.
It's not really conferring a new property. If there was a new property that made it weaker, it would be a gain of function. But there's not it's not conferring a new property. It's weakening existing properties.
Okay.
Right?
Yeah, I agree. It's not a gain of function, though.
No, because you have to have a new property. It's not a new property. It's a weakening of properties that already existed.
Yeah. By the way, the third drug we were trying to think of is Tempol, which is a polymerase inhibitor, as you could guess from the name, right?
Yeah, but isn't molnupiravir a polymerase inhibitor? It's a chain terminator.
Yeah, so is this one.
So I'm not sure that I would use this in combination with molnupiravir. Unless unless it's like combining like a drug that sat in the active site of the polio polymerase and then hitting it with ribavirin into right because it targets two different aspects of the polymerase, right?
Yeah. Yep.
So
exactly.
Maybe that would be fine, but most of them just do the active site. Yeah. All right.
Goodbye, Amy. Thank you again.
We'll work on the grant tomorrow.
Everybody say goodbye to Amy. Wave. Have a good evening, Amy. Be careful.
Yeah, grant writing. My computer should be scared.
That's good. I like that. My computer should be scared. Do we know how many individuals have been hospitalized and or died through being fully vaccinated? In the whole U.S., no. There have been isolated studies here and there, and Daniel Griffin often talks about those. You can go to the Oklahoma website. They have a great public health website for tracking COVID, and they look at infections in vaccinated and unvaccinated people. But not every state does that, so we don't have a real good indication about the whole country, unfortunately. But it would be really nice to have those numbers, right? What's the evidence for autoimmune disease caused by gene therapy vaccines? I don't know what you mean by a gene therapy vaccine. Those are two different things. We're using a virus vector for gene therapy is one thing, and then as a vaccine is another. And I'm not aware of any evidence for autoimmunity being triggered by those. What's the youngest age you think will eventually be given a COVID vaccine? In 1 or 2 years, maybe? Maybe not. Here's the thing. This is how I see it going. One day, most people on Earth will have either been infected or vaccinated with SARS-CoV-2 vaccines. And then mothers that have babies will pass on antibodies to their babies. And then the babies, when they get infected, will be protected for the first 6 months. They won't get serious disease. And then they will develop their own immunity. So they may not need vaccine. In fact, I'm thinking at some point in the future we're probably not going to vaccinate anyone anymore because this is not going to be causing serious disease. And I've talked about that a lot before, so we don't need to get into it. Oral monoclonal therapy. That's a good one. Well, you could, oral administration of humanized antibodies. So what you could do is you could put the antibodies in a capsule that will pass through your stomach without dissolving. It's called an enteric-coated capsule. And then it will come apart in the intestine where it won't be digested. And then there it can get into the mucosa. They've been looked at, yeah, for sure. I have heard that dogs can be trained to smell COVID. What are the obstacles to making a technological sensor that can detect it the way a dog's nose can? So, I don't know. That's not my area. That's for an engineer to ask. Someone sent us a link to some research being done. This is on one of the recent TWIBs where people are actually doing this. They're detecting by physicochemical methods viruses in droplets. I don't want to look it up now. One of the more recent TWIBs, maybe it was a listener pick. And so that, yes, it can be done. I don't know the details, though. I don't know the obstacles. But it's a different field from virology. When you say no one is safe until everyone is safe, is this practical? Well, that's Daniel Griffin, right? A lot of people are not going to be vaccinated, and they will eventually be infected, right? So that will bring us population immunity. Unfortunately, a good number of people will die depending on what ages they're getting infected and so forth. And that's unfortunate. I really would rather not have people dying. Is Cuba more open with their vaccine technology? I have no idea. I hear very little about Cuba. My colleagues don't talk about it very much. They do good science in Cuba. I know that much because I've heard that over the years. They have gone to meetings in the past, but I don't know about them with respect to pharma. I'm sorry. Did Brianne imply that chlamydia can almost be considered a virus? Well, she was saying that, chlamydia have to, they depend on bacteria for reproduction, like a virus does. They have to get into they don't depend on bacteria. Chlamydia have to get in cells in order to reproduce. But the difference is that they have ribosomes. They can make their own proteins. They can do a lot of things on their own. They just need other factors from eukaryotic cells. So no, they're not viral, but this idea of them getting into a cell is kind of like a virus. But viruses are missing many components, and rickettsia have them. Is there such a thing as long flu after infection? We were talking about this the other day. I was talking about it with Daniel, too. With many viral infections, there are long-term consequences, right? So with West Nile virus, you can have neurological problems. So the virus can get in the brain and reproduce and cause encephalitis. And then you can have cognitive issues. You can have motor issues for a long time, the rest of your life. But there's no virus around, okay, which is like long COVID. There's no virus around. But so in a sense, those are long West Nile, right? Something we talked about a long time ago on this live stream, post-polio syndrome. You get polio, you get paralyzed. Then you have therapy, you recover limb function, and you walk fine. And then 30, 40 years later, suddenly you begin to get paralyzed again and you go back in a wheelchair. So it's post-polio. It certainly is a long-term sequela of polio infection, right? But you haven't had symptoms all those years. You had no symptoms and then it developed later. So I wouldn't say it's the same as long COVID, where you have continuous symptoms from the onset of the acute infection and then you just never get completely cured. You never are free of any symptoms. So the neurological issues with West Nile is kind of analogous to that. But long COVID has many, many different issues, not just one, not just neurological. So, and another one that I thought of, mumps virus in a certain fraction of boys would make them sterile because it reproduces in the testes. So obviously that's a long-term consequence. So some viruses have long-term consequences. Long flu. I'm sure there are long-term consequences of influenza in some patients, right? Compromising lung function. You have serious viral pneumonia and your lungs are destroyed to the extent that they can't recover. I'm sure that there are issues like that. So I don't think that this is unique to long COVID. That's the point here, right? What factors tell the immune system to continue to produce long-term antibodies to viruses at high levels long after exposure? Well, this is a good question. Condit said the other day he had an antibody test and he has really high levels of spike antibodies, even though he's only been vaccinated. He wasn't infected. He has only spike antibody. He doesn't have nucleocapsid. So I don't think we understand why some people make high levels of antibodies for a long time. Now, of course, one possibility is with EBV, you're constantly reactivating and the virus is reproducing, right? So that could account for getting boosts in antibodies. But there are clearly other scenarios, and I don't think that we understand the factors that lead into that. Will an antiviral that develops resistance, it's the virus that develops resistance to the antiviral, right? Will be useful later in a cocktail? No, it's not going to be useful. The only one that's useful is one of the ritonavir, which originally an anti-HIV drug, which helps metabolize and activate other antiviral drugs. So it's often given in conjunction, even though it doesn't have any more antiviral activities. But no, if you have resistance, that's it for that one. Putting it in a cocktail won't help, right? Because the point of the cocktail is it's harder to get resistance to 2 or 3 antivirals than 1. Can you explain how the MDA5 receptor is implicated with the mRNA vaccine? So MDA5 is a cytosolic protein, which means it's in the cytoplasm of cells, right? And it is a sensor of RNA. So it detects when there's RNA in the cytoplasm, which shouldn't be there, and it's probably viral RNA, and therefore it then turns on the innate immune response. You make interferons and so forth. So MDA5 can detect RNA, can detect the mRNA that enters cells, and it assists in actually making a good immune response. Making cytokines is beneficial for a good immune response. So I have no concerns about that with respect to cancers, not at all. This is a transient effect. Anyway, it's a few days and it's over, and there's really no implication that I can see for cancers. Question, is herd immunity better than the immunity from the vaccine? So by herd you mean from infection, right? So there have been a number of studies comparing the two, and some say one is better and some say the other is better. And I think the bottom line is, if you ask Shane Crotty, if you ask Florian Kramer, which I did in the TWIV that should come out tomorrow, they're equivalent, but immunity after natural infection is heterogeneous, more so than after vaccination. I know that Amy wouldn't agree with me on this, but for people who are recovered from infection, some of them will have terrific antibodies, some will have moderate antibodies, some will have low, all over the place, and more so than after vaccination. That's the main difference, I think. Now, the thing is, if you get if you recover from a natural infection and then you get one vaccine dose, you have the best immunity to COVID of anyone. You have antibodies that can neutralize any variant plus SARS-1 plus MERS coronavirus, because that extra boost after recovery from infection broadens your antibodies amazingly. It's the best. So if you have recovered, you should get one shot. There's no downside. What's the story of the polio vaccine leading to cancers? So the polio vaccine doesn't lead to cancers. What happened was the early lots of polio vaccines, both the injected and the attenuated, infectious attenuated ones, they were contaminated with a virus called SV40, simian virus 40, which is a monkey virus that can cause tumors in some experimental animals, but not in monkeys. It doesn't infect humans. But it was in the vaccine, and so many people thought it caused cancer. And there were actually lawsuits saying that certain kinds of cancers were increased in people who received the polio vaccine. Many millions of people received it. I've looked at the studies. There's no evidence that this virus caused any increase in cancers in people. But as you might suspect, people would blow this up and say, yes, it happened, when it in fact didn't. How do viruses become conscious? They do not. They have no consciousness. They have no sentience. They are not able to reason or think or do anything. They are chemical reactions, complicated chemical reactions. And that's the end of the story. One wonders if there are pre-existing variants in circulation given the number of infections. Yes, of course. There are always pre-existing variants. Now, Tom, in my lecture 1, I say there are 10 to the 16th HIV genomes on the planet Earth today, and that means they're already resistant to all the antivirals we have and any we will ever make again. It's already there, especially when you have a lot of viruses like that circulating. Thank you, worm on tongue, bated breath. Excellent. Someone says that gain-of-function research is too dangerous. So we spent some time, Deb's camera, talking about this in lecture session number 3 of Virology Live. I really would like you to listen to the last 10 minutes where I explain what gain-of-function research is, why it is far broader than what you're thinking and what ZDoggMD says, because ZDoggMD doesn't know what gain-of-function research is, in fact. Gain-of-function is anything that gives a new property to an organism. It can be a virus, a bacterium, a fungus, a mouse, a human, a rabbit, a cow, a pig, anything. I made transgenic mice susceptible to polio virus many years ago by giving them the receptor for the virus. That was a gain-of-function experiment. They're very useful animals. They replaced monkeys for testing polio vaccines. So please do not agree with ZDoggMD who says they're dangerous, because he or she doesn't know what they're thinking about, okay? The purpose is, well, there are many purposes. I made an animal model for polio so we could stop using non-human primates. You want to make a better beer, you can engineer the yeast to have new properties. That would do it. You want to know if coronaviruses in bats are a pandemic threat, you engineer their spikes into other viruses and give them a gain of function to find out. They're all useful experiments. And people who say they're not simply do not understand what they're about. I am happy to explain it. What bothers me is when people say to me, these experiments are worthless, and they have no clue. They have not been doing virology for 40 years and thinking about it. So please, before you tell me they're worthless and dangerous, let's have a conversation first. I'm happy to talk about it calmly. Crazy stuff. So this hamster study, higher transmissibility by aerosol, it's in bioRxiv. Let us get it peer-reviewed first, and then we'll take a look at it. I've read it. There's some issues with it. And let's get it through peer review where they may have to do additional experiments, okay? What do I think about social virology, the use of kin selection and other social theories in virology? I don't know anything about social virology. I'm sorry. I'm I'm just an experimental virologist. You could probably explain it to me, but in this forum, it's hard to do that. I understand. Thank you, Morris, for your contribution. Mazel tov. Has any paper been written confirming the original China sequence sent to us? How can we be confident it's right? What do you mean? It's been sequenced millions of times since then, and they're all highly similar. They're all SARS-CoV-2, so I don't actually know what you want them to do. Thank you, BJR2, for your contribution, and thank you, Sue W., for your contribution to our work. Listener pick of the Nova Bat special was awesome. Bats seem to be hiding everything in their caves, including the fountain of youth. Bats are cool, but remember, rodents are 40% of mammals. Bats are 20%, but rodents are 40%. Now, I know, you don't think much of a mouse or a rat. I think they're all cool. You missed TWIV's prior twice a week? What are you talking about? We still do twice a week. We missed 1 week last week. And so you're saying it's over? Come on, Debs. I'm putting up an episode tomorrow. It would normally go up at midnight. It's going to go up in the morning because I spent 4 hours streaming with you guys today and I haven't had time to edit it. I'm working on getting people to help me. Yeah, Hotez is open sourcing his protein subunit. If Amy were here, you know what she would say, Rafe, sorry. How's that going, Peter? When should a person take a COVID-19 antiviral if the virus may be long gone before any symptom? Well, it's not long gone before symptoms, unless you have no symptoms, right? So the peak of shedding, which is when you'd want to take the antiviral, no later, is at or a few days before symptom onset, if you have symptoms, of course. So if you have symptoms, you should start taking it right away. For flu, Tamiflu, you have to take within 48 hours of your first symptoms, otherwise it's not useful. So the same would be for SARS-CoV-2 or your first positive test. If you wait till you can't breathe and you have to go in the ICU, an oral antiviral is not going to help you. It's too late. Yes. This is good. A wise person once told me people who think for a living take a shower before work and people who work take it after. Some people do both, yeah. I know people who take a shower in the morning and then they get home from work. They've been out in the world. They don't want to crawl into their bed with the detritus of the world on them, so they take a shower. That makes a lot of sense. Yeah. Yep. It doesn't have to take that long to take a shower, right? I heard that other coronas have become less pathogenic over time. How could we accelerate the evolution of COVID towards that endpoint? Can we do a selection back in animal hosts? No, you would not want to do any of this sort of thing. You have to let it take its course. I don't think you want to do that because that means you let the virus reproduce extensively to do this, and that's going to be accompanied by too much death. So let's just get immunized and let the virus reproduce and reach. I think once most people are immunized, it's going to get to the point of being less pathogenic. What's the most likely reason antiviral vector vaccines are linked to blood clots? Is it because the spikes are binding to endothelial cells? I don't think it has anything to do with the spike. It might, but adenoviruses can sometimes bind platelets. So maybe it's a property of the vector, irrespective of what's encoded in the vector. I don't think we know about it. Myocardial biopsy is a thing via catheter. Yeah, I think that's fine, but I haven't seen any biopsies of heart to look at the lymphocyte question that was asked earlier. What's the latest studies about transmission vax versus unvax? There's nothing new, really. I think transmission in vaccinated people, what we saw from the data, is that it's far, far less frequent than in unvaccinated people, but nothing new on that front, I'm sorry to say. Oh, by the way, Vanity, Acapella Science did contact me. He said, what is it that you want? And I told him we'd like you to come on, talk to you about, why you do this and maybe sing a song for us. And I never heard from him again. Go figure. I'll have to nudge him. You think CRISPR will cure any diseases? Well, successes are not, we're not there yet. We haven't started. But, however, it could be used to cure HIV, AIDS, right? Because you could CRISPR out the provirus, or maybe the provirus, which is the integrated virus DNA in our chromosomes, or maybe CRISPR out the receptor. Genetic diseases, there's a muscle disease that we talked about on TWIV last week where there's a stop codon that truncates the protein. You could CRISPR it out and fix it. A lot of diseases are amenable to CRISPR for sure. But we need to do a lot of research to make sure it's safe. And because if we don't, people will complain just like they did about the COVID vaccines, right? What are some good papers on long COVID? So if you look in Daniel Griffin's weekly TWIV reports. And by the way, Deb's camera, we still do TWIV twice. Deb's camera, we do it on Thursdays and Fridays, and we do it 3 times a week. It's just that last week I skipped the 3rd time. When I'm a slacker, I admit it, but I don't like when people call me a slacker, and I'm not, which you may not have done, but I'm just complaining, Anyway, in the last few Daniel Griffin reports, there have been some good papers on long COVID. And just because you asked nicely, let's take a look. I'm going to find the last clinical update. I'm going to share it with you here. Okay, clinical update number 82 with Dr. Daniel Griffin. And, I pick this picture, by the way, that goes into each thumbnail. Some weeks I pick a smiley one, and some weeks I pick a serious one, depending on, what's happening in the world. So here, look, long COVID. This is actually quite a good paper. Incidence, occurrence, and evolution of long COVID features, a 6-month retrospective cohort study of 273,000 survivors. So there you go. That's a good one. But if you go back, there are others as well. There's no Virology live recitation, although Amy may be willing to do it. But right now she's a bit harried, so let's wait a bit before we ask her. Is Delta deadly or more contagious? No and no. It's certainly not deadly. This is now quite clear in a number of studies, even though earlier studies suggested it might be, and the CDC said it was more deadly. It's not. It's not more deadly. Is it more contagious? It could be, but we don't have the data. And you cannot figure it out from looking at the transmission or the spread of Delta in populations because it's confounded with human behavior. And studies are just starting to come out. We did 2 on TWIV in virology labs showing that Delta appears to reproduce a little faster. And if that's true, then it could be shedding earlier. And in which case that would help it outcompete other variants and perhaps push through the population faster. But as for, it being able to infect more people, absolutely no idea about that. Even though people will say it's more transmissible and it has a higher or not. No, it's wrong. Absolutely wrong. I had long COVID in February. I had COVID and long COVID, then I had a jab. I still had long COVID, then I caught it again. My long COVID stopped. Is this normal? No, Robert, it's not normal. This is, in any situation, there are always extremes. It sounds like you had an extreme. I don't know the details, so it's hard to comment on it. But I'm sorry. I'm glad your long COVID stopped. That's great, right? And why this all happened, no idea. We wouldn't be able to comment unless we saw the details. Where does the antibiotics bit come from? Well, Tom, clinicians get an idea and they want to try it. They think they're going to save the world. But there are other approaches that are, not every cancer therapy is good and working, right? So you got to try it. You have to try different things. Yeah, for sure. Why does Amy think Hotez should lose his Twitter account? I think you need to ask her. I'm not going to put words in her mouth, okay? A TWIV on mRNA vaccine candidates for HIV would be great. Yeah. As soon as we get some papers on them, we'll do it, okay? You have your blue glasses on. That's how I knew you were broadcasting from your home. Well, also the background is interesting, whereas at the incubator it's just white. Not so interesting. But I have to say, Jillian Michaels, who did the pod, she had all white background too, and she just had a pile of books on one side of her to break it up. So I'm going to have a pile of viruses on one side. I'm going to put I commissioned a new watercolor from Michelle Banks. So this watercolor here behind me, I bought from Michelle Banks many years ago, over 10 years ago. She is a painter in Washington, D.C., and I recently had her commissioned a bigger one for the incubator, which I hope to put behind me, but I don't know if it's too big or not. We'll have to see. So I was studying immunoglobulins, and I don't know the difference between FR1, FR2, and FR3. What are they? I don't know what those numbers mean. Can you give me some context of what you're talking about? Maybe somebody else knows. I'm thinking about FC receptors, but I don't think that's right. What keeps monoclonals from being identified as foreign proteins and being destroyed? Well, they're human. They're human proteins. We make our own antibodies, and they're human. Now, if you gave someone a mouse monoclonal, it would be recognized as foreign and destroyed, and it wouldn't last very long. So that's why they humanize them or use human monoclonals. So they're not destroyed. I have I skipped some questions because they're just not right. And now some of them are being repeated again. Here we go. If CDC and FDA leadership consisted entirely of virologists and infectious disease docs instead of public health officials, how would things be different now? Well, look, Laurie Garrett said on TWIV, the problem with CDC is that there are too many clinicians who are administrators running it, whereas they need to be public health people with experience tracking down disease outbreaks. So I think what you need are non-administrators. And unfortunately, the leadership is mostly administrators. And so, yes, I think you'd need a mix of virologists, infectious disease docs, and public health people with experience in tracking down outbreaks. That would be good. And, the director doesn't have to be an administrator. I'm sorry. I don't see why that has to be. Yes, I do have a Bergen County accent. I was not born in Bergen County. I was born in Passaic County in Patterson. But I spent from 5 years of age to 18 or 19 in Bergen County. So I guess I picked up the accent. That's very astute of you to pick that up. Thank you, PMB, for your contribution. Oh, it came up in your recommendations. That's great. They often recommend TWIV to me. Hey, we have this podcast we think you'd like. Well, I make it, man. What's the story with that algorithm? I don't know. I don't understand that. Thank you, Powell, for your contribution. And I answered your question before. So no way. You don't have to contribute to ask a question. It's very nice that you support our work, but not necessary. But nevertheless, I did answer it. Are there any studies on immunity, MBCs, antibodies, CD4/8, etc.? If you get the vaccine, then get infected? No, I haven't seen any good ones. There's no, not yet. I'm waiting for that. I want to see what's going on. I totally agree with that. Yeah. If antivirals are administered early, would that cause less protection? So you're asking, would the antivirals inhibit an immune response, right? Yeah, they would, depending on when they're given, right? So it's a trade-off. So if you get them early and you prevent infection, likely your immune response is going to be reduced, but so you should still be vaccinated, I would think. Where Amy is her lab at Columbia, right? I'm at home here. Yep. What would it take for Amy to change her assessment that we'll be dealing with this for 5 years? Oh, you would have to get more people vaccinated. This is the problem. And a lot of people don't want to take the vaccine. All right, so we're going to have this for a while. Not just in the U.S., by the way, but many other countries are not being vaccinated. What is original antigenic sin? Well, it is, say you get infected with your first encounter with influenza virus as a person, strain A, and then years later you get infected with strain A1 or variant A1. Your antibody response is mainly to the original variant A. That's original antigenic sin. Even though the virus that you got infected with after it is different, you still make a preferential response to the first virus. And so that's not great, right? Because what you want is a response to the A1 because that's what's infecting you at the moment. Yeah. Do you know if the influenza vaccine has been updated for this year? Let's take a look. What year are we in? 2021, right? So what is the, let's see if we can find it very quickly. The composition of flu vaccines has been updated. So here we go. Let's take a look. The magic of the internet. Here's the 21-22 recommendations. We have an H1N1-like virus, Victoria, Cambodia, H3N2, and then we have a Washington and a Phuket. And so this has changed from the previous year. And it's slightly different for cell or recombinant-based vaccine recommendations. The Wisconsin here is different. So yes, it's been updated. And I asked Florian Kramer about that, and he said they had enough data to do the updating. Not as much as they would have liked, but they do. They do have enough. Please advise about the shingles vaccine. I've had shingles treated quickly. I also had the vaccine. You should get the new vaccine because the original one, that protection does wane. Studies have shown that. In fact, we did those on TWIV some time ago. So you should get the Shingrix. And I have to get that now that I think about it. So, yes. Are there commercial tests to discriminate between vaccinated versus natural infection? I think someone has answered this, but yes, if you look for antibodies to spike and nucleoprotein, if you've been infected, you will have antibodies to both. If you've been vaccinated, you will only have antibodies to spike. That's how you can tell the difference. What's the best method for testing? I've had the deep swab done, the drive-through shallow. Well, so that the shallow is fine. The anterior nares, where you just put the swab in the anterior part of your nose, that's fine. You don't need to go all the way up here. I had that done once. That was not very pleasant. Then, of course, there's the test method, but we're talking about an antigen test or a PCR test. And you can now have rapid PCR tests where you get the It's an amplification test, nucleic acid amplification, where you can get the results in 15 minutes. The problem is that they're too sensitive, and they will give you a positive when you're probably not even, you might not be making virus. You just don't know. An antigen test, it can be done at home. The Binax Now is an antigen test. If you're in Europe, they're either cheap or free. And those can tell you if you're currently infected and making protein. And I think they're a good indicator of shedding, because you need more protein to make a positive in those. You love Wednesday nights. That's kind of you to say that. I appreciate it. I'm spending 4 hours on Wednesdays now, live streaming. Spending 4 hours with you guys. Which is fine. Heading to New York City this weekend. Are most places relatively safe? They will ask you for proof of vaccination if you go into a restaurant in New York City. Okay, so be prepared. Bring your vaccine card or a photo of it. Anything we don't want to miss. You should ask Amy about that. But I would walk around Soho. I love Soho. They have great galleries in Soho, for sure. But, you know, the Met or MoMA are amazing, too. They're little museums that are really good. If NASA were to find evidence of microbes on Mars, would it change your opinion of life existing elsewhere? Well, they didn't come from us, right? We didn't contaminate Mars and bring them there. No, I think that meant that there's life here. I mean, it could have moved around the planets, right? I just think it's such a look, this is not a question we can settle, and I appreciate other people's arguments in favor of life elsewhere. There's so many possibilities. The numbers are so huge, but I just think that when you think about how we think life emerged here, it's just such a random event. I can't see it happening anywhere else, but you're absolutely right. It could have. But I just have an opinion about it, based about thinking about where we think life came from. I have never had durable immunity to COVID pre-vaccine. Any reason to think same hold true post-vax? No, that's a good point, and that's what Amy would say, that if you don't have good immunity after infection, the vaccine may not be any different. She has made this point many times, because I say, oh, if you've recovered and get a shot, you do really well. And she said, no, you don't know that, and she's right. So you just don't know. You could try, right? I don't think there's any harm in trying. Yeah, Twiv 281, Peter Salk. That was fun. Jonas Salk's son. Really good. What temperatures can the virus withstand? Well, it does okay at 37 Celsius. It probably does okay at 39, but once you get over 40 Celsius, it will inactivate more quickly. Yeah. This New Yorker gal gets Amy. Good. I think a lot of you get Amy, yeah. A lot of you appreciate her. And others less so. I think that goes for many people, right? I get lots of hate mail, not, because people don't like what I say. I think if they knew me, they might not do that. If you want to learn about oncolytic viruses, the last lecture of my virology course, which is up, or which you can wait for the live stream version, we talk about those as well. You would like to understand why different adenovirus vectors are used for some vaccines. Well, first of all, they're made by different companies, and they all need to have their own intellectual property, right? Otherwise, they can't patent it and sell it. So they have to be slightly different. That's one reason, but it's not the only reason. So the AstraZeneca vaccine uses a chimpanzee adenovirus, the idea being that humans would not have antibodies. Most humans would not have antibodies to a chimpanzee adenovirus. So pre-existing immunity to the vector wouldn't impair its effectiveness. And then Sputnik, the Russian vaccine, uses two different human adenovirus serotypes, right, where you get the first shot with one and the second with another. And the idea being if you make antibody, or you have antibody to the first, and you won't have to the second. And so then other companies pick other serotypes. There are lots of different adenovirus serotypes. And the higher you get in number, the more recently they have been discovered. So that's basically why people try different adenovirus vectors. And they may also have found in their own work that some work better than others at making robust levels of proteins and therefore good, strong immunity. Why not include this Q&A on TWIV? It's not really TWIV, right? I mean, I could make a separate audio-only podcast of this. I don't know. What do you think, folks? Do you think that that would be something? You know, the reason I'm not doing it right now is because I don't have time. But I met with a guy who's probably listening from local place in New York, and he's going to help me with editing and possibly even recording in the studio one day. So that'll take a load off me. I think then I would consider doing putting it out as a pod. I could just have someone else do it, yeah. I will have to put the plaque in the background. Yeah, I will do that for sure. I have an idea. Explain CD8, 4, TLR, IgM, blah, 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 etc., and how they interact in a TWIV or Virology Live. So we have a Virology Live coming up about T cells and antibodies. Yeah, so you'll get some of that. Yep. You get a plaque, not a plaque assay, yeah. But it's cool that it's the same word, right? I like that. I've heard that people's third shot reaction is more intense. I have not heard that, actually. I've heard many people don't have any reaction after the third shot. So I think you have to be careful about anecdotal evidence such as that. If you're a long hauler, 30% of long haulers claim that some of their symptoms disappear. So you have to take that into consideration when you're thinking about a third shot. Now we're talking about plaques. Okie dokie. That's great. I don't know. I have, it could take 3 to 4 months to get here. It's okay. I'm not going to hold my breath. I'm happy to have 100,000 people. Although, as someone cynically said, how do you know they're listening? Yeah, you just don't know. Okay, so is there a risk when getting vaccinated in shoulder muscle that the vaccine may enter blood vessels, and perhaps that's why we see blood clots? You're confounding things. So the blood clots are observed with the adenovirus vaccines. The mRNA vaccines are associated with myocarditis, and there's a thought that if you got the mRNA into the blood, it might go to the heart and cause that. And you may know there's a preprint out there which shows if you inject the lipid nanoparticles, mRNA containing, into mice, intravenously, they get myocarditis and pericarditis. So the question is, how often does someone put it in your blood vessel, and does the whole thing go in? I doubt it. I really doubt it. It's hard to get a bolus of virus, a mill of vaccine into a blood vessel. So I'm just not sure that's... And Daniel, someone asked Daniel this, and he said he didn't know, and he thought we needed to look into this. But it's an interesting point. I agree. But you have to remember, the myocarditis after mRNA vaccines is pretty rare, and it's completely treatable. It resolves in a few days. So it's not a big deal. I work with gamma-irradiated SARS-CoV-2. I wonder if excess exposure may trigger some... Well, yes, if you are inhaling it, which, sure, you could be, yeah. So I never get influenza, probably because I worked on it as a PhD student, and I inhaled tons of it. And I'm immune to a lot of different isolates. Yeah, probably. 100,000... it's a great number. Of course, you always would like to have more. I can't see us getting to 1 million, but maybe I'm wrong. Can you talk about ADE? So we've talked about this quite a bit, and at the risk of having people say, you say the same things every week, antibody-dependent enhancement is a situation where you make an immune response, and then when you get an infection later, the antibodies bind the virus, but they don't block infection. They help it get into cells that it wouldn't get into, and so the infection is worse. So that's where you get the antibody-dependent enhancement. This mainly happens with dengue. If you get infected with dengue 1, you make antibodies. Then if you get infected later with dengue 2, the memory response makes antibodies to dengue 1. They bind dengue 2, but they don't block infection. They lead to better infection of cells. You get worse disease. There's no evidence that this is an issue with SARS-CoV-2 vaccines. No evidence, 0. So rest assured. What do you think of LA requiring a vaccine mandate for indoor venues except for grocery stores or pharmacies? You know, this is a controversial topic. Do we have vaccine mandates we have vaccine mandates for kids to get into school, so they get them early in life. So I think at some point we should just vaccinate kids and then forget about the adults. And maybe in this point in the pandemic, it's not a bad idea because there's still a lot of infections going on. In New York, you need to be vaccinated to get into restaurants. You need to be vaccinated in many schools. I really don't have a problem with that. Why? Because I'm vaccinated and my family is, and it's fine and we're protected. So I don't see the issue. If you have an issue, why don't you talk to me about it and tell me what your problems are? If it seems likely they approve a J&J booster, should I take it or try to hold out for heterologous dosing, mRNA on top? No, if you decide you want a booster, take the J&J. The data look good. It really improves the efficacy. You know, the one shot is probably not enough. Yeah. I wear my mask everywhere still. I have to wear it on the train. I have to wear it on the subway. So I just wear it between everything. Of course, in my incubator, I don't wear it because there's nobody else there. But when I leave, I wear it. I walk on the street with it, although many people are not walking on the street. I don't want to take it on and off, mainly, because I come out of my incubator 2 blocks, I go into the train. So I just wear it all the time when I'm out and about. Can we use a nose rinse? No. A nose rinse is transient, right? So you can rinse and you'll be protected depending on what's in the rinse for an hour, a couple of hours at the most, and then it goes away. So I don't think it's useful to do that. All right. Does vaccine efficacy against severe disease wane after 8-ish months? No, it doesn't. That's the whole key here. That's why I don't think we need boosters, because vaccine efficacy against severe disease and death is not waning at all. At least for the mRNA vaccines in the studies that I have seen here. So no, every vaccinated person isn't at risk for severe disease. Now, the logic in the U.S. is that maybe vaccine efficacy against severe disease will decline. Or there's no evidence that it is, but maybe it will. So let's be prepared. That's what's driving the booster narrative. I don't buy that. I think you have to be driven by the data, and so this data that severe disease is still prevented, that's fine. Now, I understand that if you get a mild disease and you're fully vaccinated, you may develop long COVID. Let's see. Let's see if we actually do get long COVID for a long period of time, not just a few months, and then we'll talk again. I think that is a good consideration to make. Have you read the articles about the bark of quill trees that could be used for the next COVID? No, I have not. Sorry. TWIV should be added to the Library of Congress. Well, you know, NSF used to have a website, 360.gov or something like that, where they archived all the podcasts, and then they canned it. They threw it out during the previous administration, I think. But I think there are many ways that you could archive TWIB. I'm looking up this quill tree. I'm really curious about that. The tree that could help stop the pandemic. Oh, in the Atlantic. Who wrote this? Brendan Borrell. It takes you 3 paragraphs to get to the actual crux because they have to make it suck you in. All right, saponins. What does this do? Blah, blah, blah. It's expensive. Yeah, chemotherapy is made from other trees. Oh, come on. It's taken me 2 pages to get to the punchline. I don't know what it is. I don't know what there's something to do with it, but it takes too long to get to it because, the Atlantic, they have to ease you in and tell you about the people who do it and what kind of rings they're wearing and their shirt color and all this. That's how you write science communication, apparently, these days. Can you determine the number of spike proteins per sample via CT? Sorry for that little grumpiness. That I want to find an art. That's why I love science articles. You got an abstract. They tell you what you need right up front. And if you want the details, you can go further. Can you determine the number of spikes per sample via CT? No. Because the CT is just measuring all RNAs, not just spike RNA. So you can't do it. Does a COVID infection added to flu get sneezed into the room with new symptoms from the recombinations? Yeah, it could be that if you got infected with both SARS-CoV-2 and influenza virus at the same time, you would have a disease that's slightly different from both of them. And we see this for multi-microbe diseases, multi-virus infections often, that the combination disease is different from the single diseases. Yeah. If last week I was in a coma and woke up today, so you've been in a coma for a year, what are the most important things I should know about the pandemic? You need to get tested and you need to get vaccinated. I've often told Amy that TWIV is my gift to humanity. I never thought about it enduring. I figured once I was gone it would end and everybody would forget about it. But I don't think in terms of history. That was a good point that one of you made earlier, that you can recount the pandemic just by listening to older episodes. Yeah, that weighs heavy. I have to be, really careful now. I am careful. So, to what degree does heavy alcohol consumption affect vaccine efficacy? So, one of the recommendations before you get vaccinated is to cut the booze because it can interfere with immune responses. Get some sleep, yes. This precise mechanisms, not clear. How does rabies virus get in? And get into what? Usually you get bitten by some animal that has rabies virus infection, right? Bites your hand or your leg or whatever, and the virus, the teeth puncture your skin, the virus gets into the subepithelial tissues and it enters your blood system and then circulates and eventually gets into your brain and spinal cord and gives you the symptoms of rabies. Please explain how human papillomaviruses can cause cancers. Well, what happens is these papillomaviruses are reproducing in cells. And one of the things they do, they have a few genes encoded in their viruses that make the cells divide so that the DNA replication machinery is available for the virus to reproduce its genome because it doesn't encode a DNA polymerase. And sometimes by accident, those genes get integrated into the DNA of the host cell and all the other viral genes are gone so the virus doesn't kill the cell. And then the cell keeps dividing because that's what those genes do. And as they divide, they accumulate mutations, and eventually they have enough mutations to become a cancer. That's, in a nutshell, how they cause cancers. And there's more in the course, virology course. We have a lecture on how viruses cause cancer if you're interested in more. We have a lot of non-COVID questions. It's great. Can we create an mRNA vaccine for a DNA virus? Sure. The mRNA vaccine just codes for a protein. It's a carrier to bring the coding for a protein into a cell, and the cell will make it, and then you will develop immunity. So yeah, you could. You could do that, and I'm sure we will see some. Can you submit? You can submit TWIV to the U.S. Library of Congress. Yeah. So what I was going to say is I archive all the TWIV episodes, all the raw footage and the final stuff on hard drives, and I have two. I have a bare hard drive, and I put it on. Then I back that up to another hard drive, so I have two hard drives, and I'm going to keep one here and one in the incubator. It seems like overkill for something that very few people care about, but I have like 50 bloody hard drives here that I do this on. So I could submit it, but then you're always going to have to update it, right? Do SARS virus shed any components prior to entering the cells? If not, are there any viruses that do? So one of the ideas is that part of the spike is shed, right? So this S1 subunit, it binds the receptor, it falls off, and then the fusion protein is exposed so that it can infect cells. I'm not aware of other viruses. Probably other coronaviruses do that, but not other viruses, other families. What kind of virology research would you consider to be unethical? If you took a virus genome and inserted a toxin, a lethal toxin like diphtheria toxin or anthrax toxin into it, and tried to make a weapon out of it that way. There are a lot of other experiments I could think of that would fall into that category to weaponize virus. Now, I don't think they would work very well, but I don't think doing that is going to tell you anything about how the virus reproduces, how it causes disease. So those are useless experiments that are only meant to be dangerous. And so, yeah, there are some that I think we shouldn't do. People say I'm letting everything happen. No. First of all, I don't have any say into what virus research gets done. But that's the kind of research that I wouldn't allow. I'd like a whole episode on all the oops that have happened in labs with bacteria viruses and how they put into place safeguards. We could do that. We could get an expert on who could talk about that, who is all into the safety. Okay. Why are England's cases at such a high plateau? Because summertime, right? The beginning of the summer, people started doing things, getting together, demasking, and a lot of unimmunized people still. And that's driving the outbreak, for sure. If one is 2 times vaccinated with high but declining Ig titers, if they are exposed to SARS-2 but do not experience infection, can that still induce a tertiary immune response and increase titers? No, if the virus is not reproducing, you won't get a boost. No, that won't happen. You need to have some virus reproduction. The virus does not cross the blood-brain barrier. These symptoms are caused by inflammation and hypoxia. All right. There's no good evidence that and we had a neurologist on TWIV, right? Neuro-COVID, neurologist from Columbia. She said all the symptoms are likely inflammatory and hypoxic. There's no good evidence that the virus is getting in and reproducing. You don't need to get in and reproduce to have symptoms. It can be caused by other soluble factors. Yep. No, Amy cannot start earlier. She has work to do. And I'm sorry. I know you would like to have her here for the full 2 hours, but she can't do that at the moment. Maybe someday when she can get better funded. How is it going currently? Biostats master's student with biochem. What skills would be useful in preclinical virology work? I don't know what you mean by preclinical because I have a specific knowledge for that. But I think you need to have biochemistry and molecular biology for knowledge. Maybe that's not what you consider a skill. But get in a lab and do some experiments with viruses and do plaque assays and grow viruses. Don't just do bioinformatics and PCR reactions. Thank you very much for your contribution. Really appreciate it. Anything on cross-vaccination? Nothing yet. I have not heard anything. We have 635 people in the chat. Can you get me over 300 likes? There's a little like button at the lower right-hand part of the video. You bet. Yes, people are very calm tonight. It's good. They're not always that. I'm trying to be calm too. I'm trying to let things bother me less. What do I think about Dr. Collins from NIH resigning? I was never a fan of Dr. Collins, and I look forward to... I would like another Harold Varmus, frankly. I hope we can get one. I'm listening for people who know a lot about viruses, the kind that can kill me. Social skills aren't top of my list for scientists I trust with my life. Well said, Tom. That's a really good point. Don't pick on Amy. She really has great intentions. Will AstraZeneca vaccines eventually be recognized? Yeah, they will. In fact, I spoke with Sarah Hughes. Is that her name? God, I'm so just... The lady who developed the adenovirus vectored vaccines. Let's see. Oh my God, I can't believe I can't remember her name. It's not Sarah Hughes. Anyway, I'm sorry, folks. I did a TWIV with her. And it's coming out tomorrow. So she talks about it being licensed very briefly. I have to look this up. That would be TWIV 813. And she's at the University of Oxford. And her name is Sarah Gilbert. My apologies, Professor Gilbert. I got infected with COVID, and my antibodies lasted 5 months. I'm still protected. Well, you may not have detectable antibodies. Maybe the tests that were run weren't good enough, but you probably have low levels, or you probably have memory. You also have T cells, which protect you as well. Yeah, that's the mouse study that I was mentioning before. I have read it. I don't know what the implications are for people, right? I really don't know. Now we're getting to where Amy leaves. I'm a little bit past halfway mark of the questions. Unfortunately, we won't finish them all tonight, as usual. So we will go through now and find some super chats to thank. Thank you, Home. 4 hours of streamed virology. It is my pleasure to do this, folks. I'm happy to share my knowledge with you. I'm glad you think I have something to share. Because if you weren't here, well, then it wouldn't be worth sharing. Boy, you went way over 300. Thank you so much. Thank you so much. What's the mutation rate? Okay. Oh, that's Jimi Hendrix there. I don't know what you want to know. The mutation rate for me is the polymerase error frequency. And I don't have the number in my head. I'd have to look it up and tell you next week. Highly vaccinated Singapore sees a COVID spike. COVID, first of all, they're highly vaccinated, but they're still unvaccinated people. And infections alone don't matter. What matters is severe disease, and they're not spiking, and nobody's going in the hospital. So it doesn't matter. Doesn't matter. Any more? Here we go. We have one from Danny the medic. Thank you, Danny. Amy needs Red Bull, but she doesn't drink Red Bull, although medical students swear by it, right? Thank you, Jay. Really appreciate. Yeah, you should get vaxxed. 1 dose of vaccine if you have natural immunity from infection. I think that's the best, and many people do, for sure. Oh, Van Gogh is in New York. Yes. Yes. I would check that out. That's cool. And how do you prove vaccination in New York City? I had a photo of my card on my phone, and that worked. So you don't need an app or anything like that. Don't you think more people would have mRNA vaccine confidence if all the past animal studies were released? I don't think so. People wouldn't even look at them. But I think everything you need to see is out there. But it really, Rizzo Bits, I don't think people would look at them. They'd find some other reason. Thank you, Jay, for your contribution. Why is it so politicized? I don't know. People want to use it because politicians want to get reelected. So whatever they can use to get reelected, they use. And they know there are a lot of people who don't like the vaccine, a lot of people that don't like masking. So they say, don't get vaccinated, don't get masked, vote for me. That's as simple as that. It's cynical, but it's unfortunate. My mom got 1 dose of Shingrix, said she will not go back. Yeah, apparently the side effects are quite severe. And the second could be worse. So I think she should get it. I think 2 doses gives you the best protection. And you could, tell her to be ready with anti-inflammatories and so forth to try and mitigate it and take, don't go to work and so forth. I really think you should do that. Let's look for some more super chats because we have to end this because you folks are all waiting for me to say goodnight. I know. All right, here's the last question. When you furniture shop with Amy, do people assume she's your wife? I don't think so. We only did it once. And we said at the outset we're shopping for an office. So I don't think so. All right, folks. Pleasure to talk with you all tonight. All 650 of you at one point. You have made my evening joyous, and I hope you have a great week. And next Monday, we have live stream Virology at 11 a.m. Eastern. And next Wednesday, I'll be back here with Amy at 8 p.m. Stay safe, everybody, and good night.